welcome everybody to the webinar today. We will be talking about the mononuclear phagocytes in human pregnancy, and I will shortly talk about cell separation strategies that might be interesting to all of you as well. I will start the first 15 minutes of the webinar by talking about how does magnetic cell separation work, and the different strategies of cell isolation that we can find with or without density gradients. Since we're talking about blood products today, I thought that would be the most relevant. And then we will see as well which are our automated solutions and why they benefit a lot with from our reagents benefit a lot from these automated solutions to get these high purity cells that we all seek for. Then Kathy will take over with a case study for today about the mononuclear phagocytes in human pregnancy, and she will really show how these high purity isolations really helped her to enable these studies about mononuclear phagocytes. So let's start with a quick overview of Miltenyi Biotec. The company started in 1989 by Stefan Miltenyi. He was a pioneer in cell separation, which is what I'm going to be talking about today. He created these solutions, which have been growing quite a lot since then. The company has grown now up to 4,000 employees with more than 50 nationalities within our staff. And what I think is more important than what Miltenyi tells about ourselves is that we have a quarter of our employees working in R&D. So we are really pushing for innovation. We didn't stop since 1989, and we continue to push forward with the innovations, not only in cell separation, but also in different areas of research. Let's start with how does magnetic cell separation work? It has 3 key steps and 3 key components. The first step is magnetic labeling, and we find here the first component, which are the microbeads. They are antigen specific, and they will label the antigens that you are interested in. On the second step, we find the next 2 key components, which is the magnet and the column. The column can amplify the magnetic field, and this allows us to achieve minimal labeling of our cells, leaving more than 70% of our epitopes free for you to use afterwards should you need to, for example, in flow cytometry. So since we have now the cells labeled from step 1, we apply them on the column in step 2, and by gravity, the sample will flow through the column, and unlabeled cells will flow through our tube, our collection tube. If we are interested in our positively labeled cells, we then take the column out of the magnet, and then we plunge down our cells, and then here we find our positive fraction. So if you're interested in the cells that you were labeling, you will have your cells in the step 3. If you're interested in cells that you were not labeling, you will have the cells in step 2. And I said that the column was important because it allows us to achieve minimal labeling, but there's another point of the column that is even more important, and that is taking control of our cells. And this is important because we can then control if we want higher purity, higher recovery, or a balance of both. And we can control this by changing the flow rate of our by which the sample is going through the column. If we see the first animation, the sample goes pretty fast down, and then we select for high purity. If we go for intermediate speed, we will select for an average between purity and recovery, so we have like a balance between the both. And if we're interested in enriching our sample and we want very high recovery, we go into a very slow flow rate, and we can then select for higher recovery. This is an example here, and we can achieve this both in manual selection, in manual isolations, or automatic. In manual isolations, we will change the type of column that we use, and in automatic isolations, we will change the fluidic systems and the flow rate of that fluidic system. So now that we know how this works, let's see what is our solutions and how it works with blood samples. The typical workflow is we get our sample. We will proceed to isolate our PBMCs via density gradient centrifugation. We will then pick our PBMCs from the density gradient, and once we have the PBMCs, then we can isolate our target cells. Here there are 2 solutions: the positive isolation, in which our microbeads will bind the target cells, or untouched isolations, where our microbeads will be targeting all the cells except our target cells. This process here depends obviously of having the PBMCs first, so the density gradient is key to get this. But density gradients have been used for a long time, and as I said, Miltenyi is very focused on innovation. So we have created another protocol in which you don't really need to do density gradients anymore. You can isolate and touch PBMCs with one of our newer kits, and then from those PBMCs, then you can use them normally with our microbeads to isolate your target cells. And as you see also here in the top, there's also an arrow going directly from the sample to our target cells. And that is because we also have some kits that allow us to isolate directly the cell of interest from our sample. And we have these kits available for positive and in-touch isolations for the different blood products, whole blood, PAFI-code, LRSC, and Leukopak. Since today we're talking about monocytes mainly, we're going to have a quick look at the different solutions that we have for these cell types. If we already have PBMCs isolated, we have several different available kits here. I've taken the liberty to add the product numbers so you guys can take a screenshot and have a look afterwards on the website. We have direct positive isolations on the CD14, for example, the CD14 microbeads and CD14 microbeads ultra pure, and then we have isolations which are untouched for pan-monocytes, classical monocytes, and CD14 and CD16 positive. On the right side of the presentation, we see the ones that are direct from our sample, and we have here our straight from portfolio from different blood products to isolate our CD14 microbeads. And I've also mentioned that we have a kit for PBMC isolation, and this is a huge leap forward compared to the density gradients because it's faster and it allows us to standardize our results. And let's see why. So our new kit to isolate PBMCs allows us to isolate in 25 minutes our PBMCs from the starting blood sample, and it has very few steps and very, very simple. We first have a sedimentation of the erythrocytes. We then collect the supernatant. With spin. We collect then the supernatant, and then we can incubate with our reagents for 10 minutes. Put it on our AutoMACS, for example, or MultiMACS, which I will talk about later, and 25 minutes later, we have the PBMCs. This process here can be automated as soon as this incubation starts, so you only really have around 10 minutes of hands-on time. This allows us to have very clear pellet with no red blood cell contamination and also no granulocyte contamination. This is untouched isolation, and for positive isolation, we have our straight from protocols, which allow us also to go very quick from our starting sample to our cells of interest. We firstly dilute the blood sample. We add our microbeads. We incubate for around 15 minutes in the fridge, and then we run the sample through our MultiMACS or the AutoMACS, depending on the instrument that you have in hand. So using this, you can achieve very high purity of your cells very quick from your blood samples, and you can really reduce the variability between the different users that are doing cycles in your lab. So here we can see the MultiMACS, and we really put a lot of attention to our instruments here because they really help, especially straight from and untouched PMC isolations, get to the next level and really standardize the different protocols. And this is important because if you go to automation, you achieve several different things. You can standardize your processes, so you really reduce human errors. You can really make them reproducible because it's always the same protocol. It's not going to change if different people in your lab or lab technicians are doing these protocols. It's more efficient because you reduce the hands-on time, and so you can work on other tasks. You can prepare for your downstream applications, and it also increases user safety, reducing contaminations and employee exposure to samples. And below, you can see from low throughput to high throughput the different solutions that we have. The first ones are obviously manual for lower throughput labs, but it really makes sense to go for instruments as soon as you get to mid throughput because you can really take advantage of the AutoMACS features. And some of the AutoMACS features are going to be listed here. It's very interesting for multi-user labs that have several separations per day as well. It can do 1 to 6 separations per run. One important feature is that you can reuse the columns. They last up to 2 weeks. So if you really run a lot of samples, and also larger samples, it's really faster and it really helps you reduce the costs on columns. It has high reproducibility across experiments and is a perfect fit for automated isolations from a wide range of starting materials. And most of our kits are compatible with the MultiMACS as well. And then if we move towards more high throughput or higher volumes, we then find the MultiMACS Cell24 Separator Plus. This instrument allows us to put up to 24 columns on it. So we can run from between 1 and 24 samples per run. And this is really good when you have either a lot of samples or samples with high volume, because it's going to be much faster than doing it manually, of course. And then finally, it's fast and straightforward. And it really, really shines with high volume samples and high number of samples as well. And so this brings me to my summary, where you can take a screenshot, maybe, if you're interested. On our untouched PBMC isolation kit on the left side, you can find a page with more information, and then you can ask us for more. And on the right side, there's our self-separation website, where you can really find any solution that you seek for. A quick disclaimer for all of you, and then I will move to Kathy, where she will be really showing you how we can benefit from this cellular separation with high purities, controlling for high purity, and this really enables the downstream applications that especially she's talking about today. Thank you very much.
Thank you, Salwa, for that really nice introduction to the Miltenyi technology. I'm going to give an example of how we use that in our laboratory now, focusing on our work studying mononuclear phagocytes in human pregnancy, using that automated high purity isolation as kind of the core to what we do. So most of us will be familiar with pregnancy, and I put this up here. We're very familiar with the many obvious changes that occur in a pregnant woman's body as she progresses from the early stages of the pregnancy right through to delivery of the baby. But some people may be less familiar with some of the other less obvious changes that occur in a woman's body while she's pregnant. And amongst those changes are things like the hormonal changes, which of course we're not very surprised about. And there's a little summary here in the corner. And the most striking one there is that human chorionic gonadotropin peak very early in the pregnancy. And that's what we're measuring when we do those we-on-stick pregnancy tests very early in the journey to the birth of the baby. But there are lots of other physiological changes that occur as well. And the example I've just got here is those changes in maternal blood volume that increases, and there are changes in the cardiac rhythm. But every physiological system in pregnancy is affected in some way. The part that I want to talk about today is the metabolic changes that occur in pregnancy, and then how these impinge on the immune function that changes in the pregnant woman as well. So lots of metabolic changes occur in pregnancy. There are changes in the way the mother uses those carbohydrates and lipids and proteins she's taking from her diet, and she's changing the substrate utilization of those to support the growth and development of the fetus, but also of the placenta as well. And in the early stages of pregnancy, we see this initial anabolic phase when it comes to metabolism. And the mother's laying down stores of substrates and energy. So pregnant woman's eating a little bit more and laying down lots of deposits of substrates, particularly within the adipose tissue within her body. And then we have a later catabolic phase where the mother now starts to break those down, all those components she's stored in the adipose tissue, and she's breaking those down to meet the energy demands of both herself and the placental and fetal development that is really ramping up in that second half of pregnancy. And this is a little cartoon summary of some of those changes that are going on. On the left, we've got what's going on in very early pregnancy. And on the right, we've got the changes that are occurring as the pregnancy progresses. And so we've got our key three metabolic tissues there, the liver, the skeletal muscle, and the adipose tissue. And in early pregnancy, there's not huge differences to what is going on in the non-pregnant woman, but we can start to see that increased uptake of fatty acids, particularly into the adipose tissue and the laying down of those lipid stores. And we see amino acids predominantly going to the baby as the main source for the baby, but there's still glucose and fatty acids. And then on the right, we can see that change that occurs in the latter stages of pregnancy. There's less being put away and stored in the adipose tissue, and instead things are fluxing out of the adipose tissue now into the maternal circulation and down to the placenta and the fetus. And you can see that baby's not only using amino acids now, there's lots of glucose utilization by the baby as well. And of course, the baby's using fatty acids, but not as extensively as it's using those amino acids and the glucose as well. Keep pressing the wrong button. Okay, so when we're thinking about those metabolism substrates, the glucose, the amino acids and the fatty acids that we consume in our diet, what we're thinking about is their use by the cell through the processes of cellular metabolism, and therefore the production of ATP and all the biosynthetic intermediates we need to build all of the proteins, etc., that build our cells, that make our tissues, that then enable our organs to function. So this is a little summary of all those different cellular metabolic pathways that are occurring in the cell. Most of us will be familiar with glycolysis, up here in number 1, and also with oxidative phosphorylation, particularly occurring down in the mitochondria. But there's a number of different pathways all summarized on those slides. And so this is important for the uptake of glucose, feeding particularly through glycolysis there, then the uptake of glutamine, feeding through glutamine metabolism pathways and into the mitochondria and into the T-cell cycle and onto oxidative phosphorylation. And of course, our fatty acids are also coming into the cell and contributing to fatty acid oxidation pathways. And again, those cellular metabolic pathways that are centered on the mitochondria. So these pathways are then those underpinning pathways that enable the cells to function. And by having our cells functioning properly, of course, all our tissues and organs should be functioning properly as well. So this little cartoon on the slide now is sort of giving you a really overarching view of what those metabolic changes that are occurring in our pregnant women as they progress from the first to the second to the third trimester. So I've already mentioned we get that lipid accumulation early in pregnancy, and that wanes as the pregnancy continues, as the mother starts to burn those lipids, starts to break them down through products of lipolysis. And I bet if you asked any pregnant woman if she thought she was burning a lot of fat in those latter stages of pregnancy, she would probably say no, because she's obviously feeling quite large, because the baby's growing and the placenta's growing, but she's now burning fat really like a marathon runner, basically. And so we see changes in glucose tolerance that wanes as pregnancy progresses, and we see the emergence of insulin resistance as a normal feature of pregnancy. And if this insulin resistance is accelerated, we see the emergence of gestational diabetes. And of course, this all restores itself back to pre-pregnancy states very soon after the pregnancy is finished. And so we've been starting to think about that link between those mandatory metabolic changes that I've just described to you and the immunological changes that are also occurring as a really important part of pregnancy, ensuring that the mother doesn't immunologically reject the fetus. And many of you will be familiar with this concept now of immunometabolism. So this is changes in cellular metabolism as determinants of immune cell fate and function. It's really just those cellular metabolic pathways I've just described and those working in immune cells, such as the monocytes that I'm going to talk about today. And there's a really big literature now that looks at those immunometabolic adaptations by our T cells, etc., and find that these are linked to changes in cytokine production, the ability of cells to phagocytose, the proliferation of cells, reactive oxygen species production, susceptibility to death, and a whole host of effector functions. As I've already alluded to, much of this research is in T cells, but we do see quite a bit of it in macrophages, although admittedly they tend to be murine macrophage studies, and we don't see a lot in humans. And so we've been asking if there's that intimate link between those metabolic changes in pregnancy, which are really well known and well described, and maybe these are the key drivers of the immune plasticity that is a really typical feature of the pregnancy. And if we think about what's going on in the immune system in terms of metabolism, you can see on this slide, again on the left, I'm just reminding us that, there's nothing special about the cellular metabolic pathways that are occurring in an immune cell. They're just as I described on that earlier slide. We've got glycolysis, oxidative phosphorylation, and the metabolism of glucose, amino acids, and fatty acids, just like as in any cell of the body. But what's going on in the immune system is shown on the right side of the slide. We've got a whole host of other endogenous factors that are going to impact on those cellular metabolic processes. And the example on the little cartoon there are thinking about the interaction with cytokines in the microenvironment. If you're a T cell, what happens after the T cell receptor is triggered by MHC and peptide? And if you're a monocyte, which I'm going to talk about today, what happens after you get innate triggered through the pattern recognition receptors via pathogen-associated molecular patterns? And we now understand that metabolic flux that occurs at this time is fundamental to ensuring the appropriate downstream response to those signaling processes. And so if we now start thinking about what those immune changes are in pregnancy, there's many, and I don't want to go into an exhaustive list of those today, but I thought the best one to use as an example, given we're talking about monocytes and macrophages today, is this change in pro-inflammatory and anti-inflammatory cytokines. So you can see up the top, we see changes in both the T cell production of Th1 and Th2 cytokines, but more broadly, we see a flux in the pro-inflammatory versus the anti-inflammatory profile of a pregnant woman. So we follow the red line first, which is the pro-inflammatory process. We start here with sort of, prior to pregnancy, we've got the menstrual cycle there in the bottom left corner of the graph, and we see fluctuations in inflammatory processes during that time as well. And then if a woman becomes pregnant, we see a heightened pro-inflammatory response very early in the pregnancy. And this is actually a really vital part of ensuring the success of that early stage of pregnancy, ensuring that implantation occurs and ensuring the placenta can invade into the uterine wall. Then those pro-inflammatory processes wane following the solid red line, and then around the birth of the baby, we'll see a massive influx or massive increase in these pro-inflammatory processes. And these are really important in the labor process. Lots of inflammatory cytokines involved in the labor process and in the triggering of the myometrial contractions that are going to expel the baby, but also the cervical dilatation that's going to enable the cervix to open and for the baby to get out. And in contrast, our anti-inflammatory processes, which are there in the blue line, you can see they're showing the opposite pattern as we would really expect to see, I think. They're low early in pregnancy, they're elevated in the sort of second and third trimester and help maintain the environment that's going to ensure that that baby is growing happily and healthily in the womb. And then they fall around labor, and then they have some upswing in the period immediately after labor. And you may have noticed there's a dotted red line on the graph as well. And that dotted red line is to indicate what might happen in adverse pregnancy outcomes. This particularly relates to what might happen in a woman who has obesity during pregnancy or a woman who has gestational diabetes. They might not show that healthy natural fluctuation in pro-inflammatory and anti-inflammatory mediators and have a disruption of that then contributes to the disease pathology that they are presenting. So we're interested in what regulates that normal adaptation over the course of pregnancy, but we're also asking lots of questions around that in terms of what is going on with the health of the mother and the health of the baby. So we're not only interested in what is normal, we're also interested in how different maternal environments might drive changes in immunometabolic adaptation. I've already mentioned gestational diabetes, which is that accelerated emergence of insulin resistance, and that of course will have some profound effects on cellular metabolic processes. And we're interested in maternal obesity, and I'm going to give a small example of that today. Of course, we've just had COVID-19 pandemic. I say we've just had, we're still in the throes of it. And so lots of interest around maternal infection, which of course predates COVID-19 as well. And our particular interest in my group is looking at how mothers interact with the climate change that we're encountering at the moment and the possible effects of maternal pollution exposures on some of these parameters. Clearly, we're interested in how these metabolic adaptations are occurring in the immune system and contributing to adverse pregnancy outcomes. As I said, I've mentioned gestational diabetes, but there's also huge interest in preeclampsia and cholestasis, which is an itching disease of pregnancy associated with changes in metabolism of cholesterol. And of course, that impacts on inflammatory pathways as well. And there's also lots and lots of interest in how that maternal environment during pregnancy is shaping the development of the baby and affecting that baby's health outcomes for the future. And so we're looking at those immunometabolic adaptations and maladaptations and how they may be shaping the development of the baby's immune system in particular in terms of my group, but also how this may shape the baby's susceptibility to certain diseases once it's born and growing up through childhood and into adulthood. And so this is the approach we're taking for the studies that we do in my research group. So we do a lot of work with monocytes. We've got to think about the fact we're working with pregnant women. So there's quite a few ethical and practical constraints to getting samples from pregnant women. And we're trying to be as imaginative as we can be and think about the kinds of samples that are reasonable and feasible to collect from pregnant women. So we do a lot of work with blood. And the example I'm going to give you today is monocytes. We can't go in and sample synovial joints or any other kind of tissues. So we're doing a lot of work with monocyte-derived macrophages where we can generate particular populations of macrophages, and then trying to do some work with macrophages. And I'm going to give you the example of the placenta. And the placenta is a great organ because it gives you an offering of these primary human macrophages that you can use in the study of obstetrics and pregnancy, but you can also think about them as an accessible population of human primary macrophages, which are relatively hard to get. You can get alveolar macrophages and probably breast milk macrophages alongside with placental macrophages are the most accessible in terms of human studies. And on the right, I'm not sure how well that's projecting, but there's a kind of example of the approach we're taking, looking at that relationship between all of those mononuclear phagocytes and pregnancy. We're looking at them in the circulation, we're looking at them in pregnancy, we're thinking about the effects of obesity in pregnancy, so we also think about the effects of obesity in the non-pregnant woman as well as men. We're starting to try to do some work with adipose tissue, thinking about women who have cesarean sections. We can get the adipose tissue from these women, and there's already people working with this and trying to understand what's going on in that adipose tissue environment. And then in terms of what's going on in the uterus in pregnancy, we have a tissue called the decidua, and the decidua is the modified endometrium. So the endometrium would normally be shed if a woman is not pregnant, but if she's pregnant, it undergoes a process called decidualization, and then it's exquisitely positioned to support the implantation of the embryo and those very early stages of pregnancy. But we still have it in the latter stages of pregnancy, and it's a relatively accessible tissue. I did my PhD on decidual macrophages, for example. And again, we can look at this in different scenarios, and our focus on this slide is particularly obesity. And then we have the placenta itself, and there's immense interest in the effects of maternal obesity and other maternal environmental factors on the placenta. We've seen some papers on COVID-19 responses in the placenta and lots of work around obesity. And again, you can see that approach taken there, taking samples from women of different BMIs, lean women and obese women, and comparing the function of the placenta more widely, but we can go in and isolate different populations as well. And so this is now summarizing the strategy that we take in my laboratory. So we've already seen a little bit of this from the work Selva presented. So we take our whole blood samples and we do some work with whole blood, and I'm going to give you one particular example of that. We then make mononuclear cells. We're still on density centrifugation, but we are at the moment trialing the untouched PBMC kit from Miltenyi to make sure that we can swap it out with the density gradient centrifugation that has been the historical method of choice. This gives us those mononuclear cells, or those peripheral blood mononuclear cells. And then we can take those mononuclear cells and we can do something with them as well, depending on what the study is. But more often than not, we will take those mononuclear cells through to our MultiMACS, and then we will isolate CD14 positive monocytes from them. We use a positive selection strategy that Selva's already explained to you today. We have explored negative selection strategies as well. And negative selection strategies are perfectly fabulous if you're using not pregnant samples and children and all kinds of things. But if you try to do them on pregnant women, you get way too many platelets carried through. So we typically deploy a positive selection process. So we can take our monocytes and then we can use them for lots of different downstream applications. And the examples I'm going to talk today, I'm going to give you an example of mass spectrometry, an example of extracellular flux analysis, and an example of transcriptomics on the nanoString, and I'll explain each of them when we get there. We can also take those monocytes and under defined cytokine environments, turn them into macrophage populations, because there's lots of preceding work in the literature that says if you expose the monocytes to cytokine A, B, and C, they will develop into macrophages that behave like a particular type of tissue-resident macrophage. And I'm going to talk about one population in particular. And of course, the other thing we do is we take our placenta and we can mush our placenta up, we can enzymatically digest it, we can do our density centrifugation, and we can take through to our macrophages in this case, using the same CD14 microbeads that we use for the blood and the monocytes. So thinking about this journey, that's what I'm going to take you through for the remainder of the presentation this afternoon, this morning, wherever you may be in the world. So thinking about our whole blood strategy, there's lots of fabulous things you can do with whole blood, and you don't have to do any downstream processing. You can go straight to whole blood flow cytometry. And the example I've got here, and it's a kit we find highly valuable in the lab, is the 8-color immunophenotyping kit. We can use an automated hematology analysis approach, and that will give us information about our monocytes, our neutrophils, our eosinophils, our lymphocytes in total, as well as our basophils. but it won't give us any indication of the lymphocyte subsets, our monocyte subsets, that might give added value to the analysis we're doing. So here we can use whole blood, we can use the 8-color immunophenotyping kit, we can use a really well-defined flow cytometry gating strategy that's shown on the slide here, and then we can go in and quantify all those different cell populations you can see listed on the left-hand side of the slide there. And then we can compare those in our different populations. And we've used this to look at pregnant women who have high BMI versus pregnant women who have a low normal weight BMI. And we've done this in peripheral blood that we've taken from women when they're 28 weeks of pregnancy, and we've only used women who are negative for gestational diabetes. And so we can find lots of fabulous differences in the peripheral immune cells of women with and without obesity at 28 weeks of pregnancy. And I put a few examples up here. I didn't want to go into too much detail. This paper has just been published, so if you're interested in finding out more about it, you can follow up the publication as well. But the 3 examples I've got here on the slide, the top left one is looking at that T-cell ratio. So using the immunophenotyping kit, we can enumerate the CD4 and CD8 T-cells, and we look at changes in the total numbers of those. And we can also calculate our CD4 to CD8 ratio. And you can see there's a dramatic change in that with the mother's BMI. There's a decline in CD8 cells in particular, and so the CD4-CD8 ratio. And this has been described in not pregnant people with obesity as well. And similarly, up on the top right, we can look at our NKT cells, and we can see that nice decline that occurs in NKT cell numbers in the circulation of women as their BMI increases during that pregnancy. And again, this is something that's also been shown in not pregnant people as well. On the bottom panel there, we've got our monocytes, a little bit less startling than the top 2 observations, but we can able to go in and subset the 3 different types of monocytes, the classical monocytes, the intermediate monocytes, and the non-classical monocytes, and look at those 3 different subsets which are differentiated by their CD16 expression. And you can see that in the case of the intermediate monocytes, which are the little blue squares, there's a sort of a slight decline of those with increasing maternal BMI. And we've done a whole pile of extra work on these samples, and we've gone on and shown lots of different changes, and those are summarized on these slides here. We've looked at circulating parameters, shown that there are changes in things like leptin and interleukin-6 and MCP1 or CCL22, I think it is. These are increased in the circulation of pregnant obese women, just like we would see in non-pregnant obese people. I've highlighted that CD4, CD8 and NKT cell changes, and we see changes in those monocytes. We see the decline in the intermediate population, but overall in our monocytes, we see a decline in the mitochondria, and we see changes in various surface markers on these cells using a whole range of flow cytometry, and we can see changes in the Th1, Th2, Treg, Th17 balance in these pregnant women. And what we see are a lot of things that are reminiscent of what we see in non-pregnant women, but we've now confirmed that they're occurring in pregnant women. And again, if you're interested, I refer you to that paper that's just been published in Clinical Experimental Immunology. So that's thinking about our whole blood approach. Now we're thinking about our mononuclear cells, and we're taking them through using our CD14 microbeads that Selva introduced you to, using the methods he introduced you to, and positively selecting our CD14 positive monocytes, and we get nice purity. We would run at about a 92 to 95% purity of the cells most days in my laboratory. And so we can use these for a whole host of downstream analyses, and I'm just going to walk you through a few examples of those different types of approaches that we take. So the first one is just, I don't want to say straightforward, simple flow cytometry. I love flow cytometry. It's a great tool. It's one of the key tools that we use in my laboratory, but we can do lots of powerful things. Of course, we can do this in our whole blood, and I've already given you an example, and we can do it on our mononuclear cells, but because we're ultimately interested in potentially subsetting these based on their CD16 expression, we've gone with using our purified monocytes for much of this analysis. But again, it depends on the studies we're doing. So here we're just looking at CD16 expression of those monocytes that we've isolated. And here we're now moving into studies of women at term pregnancy. So these are women just a few days to a few weeks before they have their babies after 37 weeks of gestation. And so we can use CD16 to stain the cells, and then we can see there are differences in the relative abundance of those 2 major monocyte subsets, the CD16 positive non-classical monocytes and the CD16 negative classical monocytes. And we're not the first to describe this, we're confirming previous literature, but pregnant women have an increase. And if you look at different stages of pregnancy, you will see these increasing as the pregnancy progresses. We can also look in more detail at those cells using other tools. And the examples I've got here is the flow cytometry through the middle of the slide there is using MitoTracker to look at the mitochondrial content of our monocytes. And here again, we're using CD16 to identify our classical and our non-classical subsets. And then we're using MitoTracker to provide a quantitative measure of the mitochondrial mass in those monocytes. And we've got the little black circles there are our not pregnant women, and the little blue squares are our pregnant women. And on the left, we've got our non-classical CD16 positive monocytes, and on the right, our classical. And you can see that in both populations of our monocytes, we see a decrease in the mitochondrial content in our pregnant women. So to us, that was a really interesting observation. And we were able to confirm this by taking those really nicely purified monocytes and doing some mass spectrometry on them. We were able to do mass spectrometry for cardiolipin, which is the graph on the bottom there. Gray is again our not pregnant, and blue is our pregnant women. And you can see a nice difference in the cardiolipin content of these cells as well. And cardiolipin is a lipid that's particularly expressed within the mitochondria. And so this is confirming the fact that these monocytes from our pregnant women have a reduction in their mitochondrial content. So having seen a reduction in the mitochondrial content of our monocytes, using a strategy that's really enabled us to look at the same cells for their MitoTracker expression and take them through to mass spectrometry as well to do the cardiolipin analysis, we then wanted to look at the functional responses of these cells. So the methodological approach we then took was to do extracellular flux analysis. Anyone who is familiar with this might call it seahorse analysis, which confuses people. People are always a bit surprised when we use the terminology because they know we're a human immunology lab, and they get very confused when we mention seahorses, and we have to explain it's just the slang name for the piece of equipment. But this particular piece of equipment allows you to actually measure oxidative phosphorylation and glycolysis in intact cells. And we do this on our monocytes, as I'm showing you here today, but we do it on T cells and eosinophils and neutrophils and all kinds of immune cells. And this enables you to do to inject the cells with a whole host of different inhibitors and activators and uncouplers of the different metabolic processes within the cells. And you can see the little table on the bottom left has got the 4 key drugs that I'm going to be using in the assays I'm talking about today. And we can inject these in different sequences, and then we can measure what's called the oxygen consumption rate up on the top right there of the slide, or we can measure the extracellular acidification rate, which is in the bottom right there of the slide. And the oxidative consumption, which we use the shorthand of OCAR, allows us all kinds of measures of oxidative phosphorylation. And you can use different points on that graph that trace that is generated, and you can calculate different things such as spare respiratory capacity, maximal respiration, and a whole host of different parameters. And then equally on the bottom, you can do the same thing, but here you're measuring extracellular acidification rate, which is a measure of glycolysis. And again, you can look at various glycolysis parameters using the data that you generate in that trace. So we took our monocytes that we purified on the MultiMACS, and we were able to put them into the plates for the extracellular flux analysis. And then we can generate those oxygen consumption rate traces and those extracellular acidification rate traces, as I've given you the example on the previous slide. So that's what's now shown on this slide. And again, we've taken monocytes from our non-pregnant and pregnant women at 37 plus weeks of gestation. And the most startling thing you'll see is the oxidative consumption rate. Again, our pregnant women are in blue, and you can see they have a very reduced oxidative consumption rate, suggesting they've got reduced oxidative phosphorylation. And you can see that summarized in the graph below as their respiration rate calculated from that trace above. And you can see that maximal respiration rate is different, significantly reduced in our pregnant women. And on the right, we've got glycolysis, and we can see there's not any real significant difference in the glycolysis that's occurring in these cells. And to us, of course, that made sense. We knew our monocytes had less mitochondria in them, so it was perhaps not surprising that when we looked at cellular metabolism, focusing on oxidative phosphorylation and glycolysis, it was only oxidative phosphorylation that showed that decline in our pregnant women. And then we wanted to see what the functional consequences of that were for those effector function that the cell is doing. So we know we've got that fewer mitochondria, We know that there's less oxidative phosphorylation in these cells from our pregnant women, or what's going on in terms of an effector response by those monocytes. And we use cytokine readout to look at that. But of course, you have to be very thoughtful about what you stimulate your cells with. Lots of people would stimulate their cells with lipopolysaccharide, for example, which is what we did, and we didn't find much of a difference. But lipopolysaccharide activity is very dependent on glycolysis, and we knew that glycolysis was not dampened in our cells from our pregnant women. So we searched around for something that may be a stimulus that was dependent on oxidative phosphorylation, and we settled on muramyl dipeptide, or MDP, which is a NOD2 NLR ligand. And you can see the cytokine responses from our pregnant and not pregnant women on the right, and we can see reduced interleukin-1 beta, significantly reduced interleukin-6 and TNF-alpha in the cells from our pregnant women, indicating that poorer oxidative phosphorylation, that lesser mitochondrial mass, is having that functional effect on the cells. And so we wanted to really get in and understand why this may be occurring. So we then did transcriptomics on the cell, and we used the NanoString N-counter sprint profiler. And on this, you can analyze 800 target genes on your cells. You only need about 80,000 cells. We chose the N-counter metabolic pathways panel because we're interested in cellular metabolism. And in our case, this was particularly beneficial because we didn't have to extract RNA. We could make cell lysates, and meant we could increase the workflow, and we could also ensure that we were getting good fidelity of our assays just on those very small numbers of cells that you can use there. And the little cartoon on the right there is just the workflow for how the NanoString works. And so then the kinds of readouts you get there are those things you see in all the papers on transcriptomics. You can take this volcano plot on the left here, which is gene by gene analysis. And here we're looking, on the right of the volcano are things that are higher in our pregnant women compared to our not pregnant women. And on the left, we've got those things that are lower in our pregnant women versus our not pregnant women. And we can see there's lots of genes of interest that have come up, and we're pursuing those at the moment. But we can also take a sort of more heat map approach, and we can see that heat map analysis on the right here. We've got our not pregnant women on the top of the heat map, not pregnant 1 to 6, and then our pregnant women down the bottom. We've got our different pathways along the y-axis of the heat map, and a blue is an increase in expression, and the amber is a decrease in expression. And there's lots of different pathways in this metabolic pathways panel that are up and down regulated in our pregnant women. These include amino acid synthesis, mitochondrial respiration. And so again, they're helping us understand what's going on in those pregnant women. So just to summarize that before we get to just a few examples of the monocyte-derived macrophages and the placental macrophages. So we showed that there was an increase in the non-classical monocytes in pregnancy, confirming what was already known. But we were the first to show this reduced mitochondrial content using our flow cytometry and our mass spectrometry of our really highly purified monocytes. We've showed reduced oxidative phosphorylation, and that this is associated with decreased response to a pathogen-associated molecular pattern, as long as that PAMP is dependent on oxidative phosphorylation. And we can see relevant changes in our metabolic pathways using that NanoString-based transcriptomics. So immunometabolic adaptation is definitely occurring in the monocytes of these pregnant women. And we're now trying to understand why and sort of the timelines of this during the pregnancy. And so some of the other things we're doing as well, and I touched on this earlier in the presentation, we talked about the monocytes in the blood, but I just want to give you a couple of examples in the last few slides of monocyte-derived macrophages and our macrophages from our tissues. So this is the only example of our monocyte-derived macrophages. We're looking at monocyte-derived osteoclasts in particular. And here we're taking out our nicely pure monocytes. We then expose them to that defined cytokine environment, which in this case is MCSF and rank ligand. And we culture the cells in this for 11 to 14 days. And over that period, they very nicely turn into these highly syncytialized osteoclasts, the macrophages that we would find in the bone, for example. These are the macrophages we would find in the bone. And you can see the confocal microscopy along the bottom there. At day 0, we can see our beautiful little monocytes. That little, the red is the cytoplasm, or the plasma membrane, and the blue is the nucleus. And you can see the nice kidney shape of our monocyte nucleus. And then over the course of the culture, in the presence of those two cytokines, they start to merge these different monocytes, and they form these multinucleated giant cells, which you can see in the other images there. We can see cells with 6, 7 nuclei inside them. And then we're staining them with trap antibody, which is antibody for an enzyme that this particular cell type produces, which helps it degrade the bone. Then we've got TOM20, which is a mitochondrial stain, so you can see those green mitochondria clustering around the nuclei. And then we've got the rank antibody, which is the receptor for the rank ligand we use to differentiate cells. The very last thing I want to do is talk about that placenta. We get that placenta, it's a natural waste product after the baby is born and normally gets chucked out by the clinical team. So we acquire that and then we take it and we process it through for macrophages. And you can see the fetal side there on the left, which has got the umbilical cord coming out, which of course will be attached to what becomes all of our belly buttons. And then we've got the maternal side down there. And we're taking a very similar approach that I've already described to you. We take our placentas, we take them from women of different BMIs, is what I'm talking about today, and we isolate those placental macrophages. And again, we use the MultiMACS to do this. And we're taking a very similar strategy to what I've described with the monocytes. So I'm not going to go into any great detail. We use the seahorse analyzer, the extracellular flux analysis, and we can see the oxidative consumption as a measure of oxidative phosphorylation. We can see the extracellular acidification as a measure of glycolysis. And it's a little bit more complicated than what I showed you with the monocytes, because here we've looked at the cells after they've been cultured in either the absence or presence of lipopolysaccharide. And what we found was that those macrophages from our obese women, and here we use women who have a BMI greater than 35, so they're quite obese by this stage, and we're comparing them to lean women, and we can see that there is a dramatic difference. The macrophages from our obese women have very reduced oxidative phosphorylation, but they also have very reduced glycolysis. So we're now trying to tease apart what that means functionally for the cells. I just want to give you one example of how we can analyze those functionally. We can use Frodo-labeled E. coli to look at the phagocytosis of these cells. Here outside, the cell pH stops the fluorescence of the Frodo-labeled E. coli, but once the E. coli is internalized and the pH of the lysosome, they're exposed to the pH of the lysosome, they will start to fluoresce and can be detected on the flow cytometers. So we can do lots of lovely flow cytometry analysis there as well. And then what we found on the right was that our obese women, again in that pinkish color, have much reduced phagocytosis, especially as you're increasing the amount of bacteria they're being exposed to. Their phagocytosis is limited, and we're now trying to understand how that altered phagocytosis is linked to those altered metabolic processes we described in those cells using extracellular flux analysis. And so just looking at this last bit of work here, looking at that placental macrophage immunometabolism, we know there's some kind of functional metabolism defect occurring in these cells. And we're now looking at how that may be associated with gestational diabetes and various other pregnancy disorders, and placental dysfunction, inflammation, lipotoxicity, the things that are actually going on in the placenta, and then thinking about what that means for the baby. And so just to summarize, there's that strategy that we've been using throughout the presentation I've given you today, where that MultiMACS high purity isolation of our cells sits right at the center, and it enables us to do all those downstream analysis processes that I've presented you today. And it also underpins our other work in our macrophage setting, both the monocyte-derived macrophages and those tissue macrophages we can isolate from the placenta. And this last slide really is just to remind you, this is really advertising what my laboratory group does. And so this is a strategy we're using to try and understand that pregnancy environment in its most natural of states, but also how it may be perturbed in response to different environmental influences, and the role of the monocytes and the macrophages in particular in that. So just on the last slide, I'd just like to firstly thank Miltenyi for inviting me along today to talk about the work we're doing on mononuclear phagocytes, and just thank the people in my team. So April Rees did her PhD on the monocyte work and recently successfully awarded that. Hamza Nasser is doing the monocyte-derived macrophage work, and Meg Chambers is doing the placental work. And we really couldn't do the work without the midwives up on the antenatal unit in the central delivery suite, and we absolutely could not do the work if it wasn't for the women who agreed to be part of our studies and let us take their placentas and their blood. And along the bottom is just some of the various people we collaborate with. Thank you, everybody.